Hello, and thank you for joining this webinar. My name is Ben Schmidt. I am a staff scientist at Physical Electronics in Chanhassen, Minnesota. Today we will be discussing scanning X-ray imaging and how it can help us with our XPS analysis. So to motivate this webinar, we have two questions that we will consider when we start an XPS analysis. The first is, how much do I really know about my surface when I start my analysis? The second is, how can I be sure that I have chosen the proper area for analysis to get reliable results? And so with those questions in mind, today's agenda has three parts. First, I will introduce the concept of SXI as it relates to PHI XPS systems. Next, I will talk about some of the applications of SXI and uses. And then lastly, look at some examples of how within our software we can use the SXI for us. To start with, I will mention that SXI is a fundamental feature of all PHI scanning microprobe XPS systems. Currently, PHI offers 4 XPS systems, the Quantera 2, X-Tool, VersaProbe 3, and Quantez. All of these have a focused monochromatic aluminum K-alpha X-ray source. The Quantez additionally has a focused monochromatic chromium X-ray source for hard X-ray analysis. And each of these can benefit from SXI. Now we will start our discussion of SXI with a general overview of the scanning X-ray microprobe used on PHI XPS systems. The scanning X-ray is composed of several parts. First, we have a Lab 6 electron source, which is used to generate an electron beam which is focused at our anode material, whether that is aluminum, chromium, or some other material. The X-rays generated from the anode are then focused by a monochromator and focused back on our sample. As we adjust the position of the electron beam, we can in turn adjust the position of our X-ray beam on the sample. We do have the ability to change the size of the X-ray beam on the sample based on the focusing and size of the electron beam on the anode. And we can create X-ray beams with diameters less than 10 microns up to about 200 microns in diameter. And we can use different X-ray beam sizes based on the feature size of our sample that we are investigating. And now, scanning X-ray imaging. As we scan our X-ray across the surface, we will actually be generating secondary electrons from the different points across the sample. And as these secondary electrons are generated, we can collect those through the exact same analyzer and detector that is used for our photoelectron analysis. SXI, as we call it here, scanning X-ray imaging, can also be known as scanning X-ray induced secondary electron imaging. And because we are imaging the secondary electrons, this gives us an SEM-like capability to do sample navigation, analysis area definition, and speed, in that we can collect these SXI images within just a few seconds. And as the saying goes, a picture is worth 1,000 words. And so with an image as shown at the top right, we have very quickly an idea of areas of interest, whether they be contaminants or features that we want to investigate. In an SXI, the primary contrast that you will see is due to the number of secondary electrons that were generated at that point as the X-ray scanned across it. And the contrast then is also partially related to the photoelectron yield of that particular material, which can highlight chemical differences. And so this enables us the ability to observe localized surface contaminants or maybe other thin pattern features that are often not visible optically. Now at a constant stage position, we can raster the X-ray beam up to 1400 by 1400 microns. We can also typically use an X-ray beam size of 7.5 to 10 microns in diameter for this imaging to get good lateral resolution in our image. And so the importance of SXI is that it provides us with a starting point to choose our analysis conditions for any type of sample. We can quickly produce an SXI, and that will tell us things like what features are in our analysis area and whether we want to investigate those by using a large X-ray beam where we are averaging over a larger area, or if we have smaller features, we can drive to those with a small X-ray beam to get very good lateral resolution. In addition, it allows us to 100% accurately choose the analysis area that we would like. Because the secondary electrons and photoelectrons are collected through the exact same analyzer and detector system, if we choose a point on the SXI, we have the utmost confidence that the photoelectrons that we generate from there will be collected at the same area. So a few of the benefits that we will use for SXI in addition, we can ensure that representative areas of our samples are being analyzed. We can quickly see non-homogeneities and then either investigate those or navigate away from them to get an area more representative of our sample. We can also look at things like thickness of coatings on a sample based on contrast in the SXI. We will also talk about how we can incorporate SXI into depth profiling and use that information before, after, and during the profile to understand our area of analysis. So the first example will be how we can use SXI for locating defects or in general areas of interest. What we see here is a coronary stent sample, and we have collected an SXI within just a few seconds on the left, and we can see that there is a large contrast highlighting the boundaries of the stent itself. We also see a dark spot of approximately 100 microns in diameter right away, and this could be a possible contaminant on the surface. What we can do is, from this SXI, place an analysis point directly on the image where we want to perform the analysis, and we can choose an appropriate beam size of, say, 20 to 50 microns in diameter, which will only collect data from that particular spot. And so a survey scan of that spot shows that we have fluorine, carbon, oxygen, silicon, and a little bit of chlorine as well, fluorine too. We can also perform high-resolution analysis of the carbon-1s peak, and it shows that we, again, as we saw in the survey, the presence of fluorine, which would be a contaminant in this case, and the high-resolution scan of carbon also reinforces this, that we see carbon-fluorine type bonding. In this example, we are investigating a group of carbon granules, and we have first collected a photo from our intro chamber of the XPS system shown in the top left. And you can see this clump of granules that are fairly uniform in their dark color, so nothing really sticks out right away. However, in the bottom left image, there is an SXI that we have collected, and we can see that a couple of these granules are easily visible, and there are some differences in the color of the two, in that one is a sort of monotone, monochromatic gray color, whereas the other one has both dark and bright features. If we wanted to choose a large area analysis across several of these particles, we could do that. However, we would be averaging the elements and the signals across that entire region, and so we may lose some detailed information as shown by the SXI, in that we have both bright and dark spots that could be interesting for analysis. What we have done in this case, as shown on the survey scans, is we have drawn areas directly on the SXI of approximately 250 by 250 microns in size, and using a 100 micron diameter X-ray beam, collected surveys from each of the two particles individually. And we can see that the more uniform particle to the right and top is composed mainly of carbon and a little bit of oxygen. However, the survey for the particle with both bright and dark features has about 11% silver in addition to carbon and oxygen. And so right away we can see that there are differences between the two particles. Furthermore, looking at the particle to the bottom left that has the bright and dark features, we can see that there are possibly some more information that we can extract if we really try to understand what is happening between those bright and dark features. And so again, we can ask the question, is this SXI contrast that we're seeing possibly related to the silver content that we saw from the average survey? And now what we're looking at is a different particle that also shows the bright and dark features in the SXI. We again are using a 9 micron diameter X-ray beam for this SXI. And as I show here, we have several fields of view that allow us to see larger areas on the particle, going from 1400 by 1400 microns down to 350 by 350 microns. And so by going to these smaller fields of view, we can very easily select points of bright and dark regions for analysis. And as you can see, the size of these bright regions are on the order of maybe 40 to 50 microns. And so we will use a 9-micron diameter X-ray beam to analyze one of the bright areas and one of the dark areas. And again, this was done by simply clicking on the SXI where I wanted to perform this analysis. As you can see, in the bright area, we have about 33% silver compared to the dark area where the silver is less than 1%. And we also see in high resolution scans between the red and the blue curves the difference in carbon and silver content. And so with this small area spectroscopy, it is suggested that the silver is localized on the carbon particles and actually correlates with the bright areas of the SXI. And so with the aid of the SXI, we are able to extract more information than we necessarily could from a large area analysis alone, but again, we had the option to do both if we wanted to. The next example is showing how we can use SXI to locate representative areas for analysis. The sample here is a Perovskite material, and in an optical image taken before the analysis, we can see that there are some obvious cracks and defects in some of the corners and outer edges of the image. However, in the central area of the image, it looks somewhat uniform. But once we put it into the system and collect an SXI within a few seconds, we can see that there are some dark smudge areas in the area that we thought were uniform. Now, if we wanted to collect data here from a large area analysis, we could do so, but we may wonder if some of those dark smudges are contributing to strange results that we may see. But with the SXI, we can choose to either investigate those dark areas or potentially move away to where we see an area that has a more uniform SXI. In the next example, we'll be using SXI to observe non-uniformities in film thickness on a polymer material. This is a polyethylene sheet, and we're seeing an optical image collected of this material. There is a lot of roughness and texture of the polyethylene substrate, and so it's difficult to see any non-homogeneities optically. However, we have collected an SXI on the right side of the window, and we can see that there are areas of bright and dark features. If we analyze one of these bright features with a small X-ray beam localized to that bright spot, we can see in the survey that there are differences in the fluorine to carbon ratio of the two regions. It appears that the bright area has a higher fluorine concentration of about 62% compared to about 24% for the dark region. And as you can see here, the two analysis points we're using are spot 1 is our bright spot, which has high fluorine, and spot 2 over here is then the darker region with lower fluorine content. So we may think that the fluorine content in the bright area is a little bit thicker in that this is a fluorocarbon coating on the polyethylene. We can also look at high resolution spectra confirm our suspicions, and the carbon 1s spectra shows that indeed in the bright area on the red curve we do have higher CF2 type bonding compared to the blue area where we see a larger ratio of CC bonding relative to the CF type bonding. Again, suggesting that in the bright area that we see in the SXI, we have a little bit thicker fluorine coating. Next, we will see how we can apply SXI to our depth profiling and mapping. One of the advantages of the scanning X-ray beam is that we do not have to move the stage whenever we want to do a multi-point analysis within the SXI field of view. And so we can take advantage of this to do a multi-point depth profile without having to move the stage. As we show here, we have an SXI of a patterned silicon wafer in which we see three regions of interest. There is a large uniform field, which is labeled 1. We also see a square pad labeled 2, and then a line labeled 3. We can select those points directly on the SXI and then perform our sputter profile analysis. After sputtering, we will collect data from each of those 3 points, and again, without having to move the stage. And so within a single sputter crater, we can collect profiles of 3 different regions. And so this can be very useful if you have limited sample space and do not have the ability to perform analysis depth profiling in multiple locations. You can collect that information all at once. And so the depth profile is shown here in that position 1 has a silicon oxynitride type profile, whereas the pad has an oxidized surface but then is a tungsten silicide material. And then the line, number 3, is a silicon oxide. In addition, while we're profiling, we can use SXI to look for non-uniformities that may affect our overall interpretation of the profile. This is an example of a diamond-like coating on steel. And we've started with an SXI on the left, and we see that it is fairly uniform. And if we perform a large area analysis across this region, we will see that we have carbon, oxygen, silicon, and some low levels of calcium, sulfur, and sodium. So based on this, we have chosen to do a depth profile. And after doing some sputtering to remove the surface layers, we can collect another SXI to see the progress. And one thing that we notice from the SXI after sputtering is that there are several bright particles that show up very easily in the SXI. And these particles are on the order of 25 to 30 microns in diameter. So we can do a point analysis on the particle to understand what that may be. And what I show here is we have done an analysis of this 25 to 30 micron particle using a 9 micron diameter X-ray beam, and we see that it actually is composed of multiple metals such as tungsten, iron, and chromium. So we can see that these metals are actually localized to the particle area. And so if we looked at a survey and detected, for example, tungsten, iron, and chromium, and then tracked those elements during our profile, we may come to the conclusion that we have low levels of metal contamination throughout the coating. However, by doing a localized small area analysis, we can see that it is not homogeneous within the coating, but simply localized to where these particles are present. And now we will use SXI as a starting point for our mapping. This is another patterned silicon wafer, and we can see that we have a bright square pad in the middle, but we also have features to the top right of the pad, which are designations to understand which pad is which. We also have some other features below the pad, some different lines and connections being used. So I've used live SXI navigation to navigate to this particular pad of interest, and I can zoom into it and assign the map area that I would like to choose, in this case shown by the white box of 200 micron by 200 microns. For the map, I have used a 9 micron diameter X-ray beam within that mapped area, and we can see that we have produced 4 elemental maps of tungsten, silicon, nitrogen, and oxygen. The tungsten, nitrogen, and oxygen correspond to different areas on the pad, around the pad, or surrounding the outer area away from the pad. However, the silicon appears to be uniform across the mapped area. And we can also extract more information from silica that I will show in the next slide. One of the advantages of the snapshot mode that we use while collecting maps is that each pixel contains a full spectrum. And so we can use a linear least squares fitting algorithm to extract spectra that have similar shapes from different regions of the map. And as I've shown here, I have drawn a box in the pad region, and that spectrum shape corresponds to a tungsten silicide. We also have extracted spectra from the bottom right area, and that would have a peak position and shape of a silicon oxide. And then the area just below the pad has a peak position corresponding to a silicon oxynitride type material. And we can take each of those individual components and then overlay those into a silicon chemical state map to show chemical bonding states on the map as well. And again, this map area was determined based on our SXI. So now that I've shown you some of the applications of SXI, we will talk briefly about how this can be used in practice. When we introduce a sample to the system, our general workflow is to first drive to the area of interest and perform a Z-align. As shown on the left, this is a platen of a silicon wafer with a coating mounted onto a sample platen with adhesive tape. I can use this intro photo to do a coarse approximation of my sample area of analysis, and then I turn on a Z-align to find the proper sample height to get the most signal. Next, I will turn on SXI, and within a few seconds I will see an image such as shown here on number 2. And in the area that I have chosen, based on my photo movement, there is a dark smudgy spot right in the middle of my viewing area. Now if I collected a large area analysis from this region, I would be collecting from both the uniform area off the smudge and including the smudge itself, which may give me incorrect results representative of this film. So with the SXI running, I can either click on the SXI to move the stage to an area where the SXI is more uniform, as shown to the top right, or I could investigate the features that I'm seeing in that smudge by zooming in on those features as shown on the bottom right. Once I've moved to the general area of analysis that I would like, I can then choose the specific points that I want to use and choose an X-ray beam that is representative of the analysis area I would like to investigate. And again, to show how this is used in our SmartSoft software, we have several controls for starting and stopping the SXI. We can also choose the field of view, again up to a maximum of 1400 microns. We can change the refresh mode from a slow, fast to total, in that fast will refresh about once a second, so it's very good for navigation while we image the SXI live. And in total, we can then collect a few frames to clean up the image. And then once we have an SXI, we can save it, and as shown on the right side, we can simply draw points, lines, and areas that represent the analysis region that we would like to investigate. So to show how this can be done in real time, I have a short video of looking at a sample of a metal foil. And this is a representative window of our SmartSoft software. As you can see in the top left, we have a photo that was collected in our introduction chamber. This is a metal foil mounted on our sample platen. And from the photo, it looks fairly uniform. So once it has been introduced to the chamber, the first thing we will do is perform an automatic Z alignment, which will adjust the sample height to get the most signal into the spectrometer. And we can track the progress here. As you will see in the figure, the Z position is shown on the bottom axis, and then the count rate is shown on the y-axis. And so we will slowly move the stage up until we notice a change in the intensity, and then we will back down and take smaller steps to find the optimum sample height. As you can see here, the intensity is increasing as a function of sample height. We have reached a peak, and we are starting to see a decrease as we move past our optimum position. So the stage will then move to that peak position where we have our optimum sensitivity. Now we will turn on SXI. We currently have a 1200 micron field of view in this example. And within just a few seconds, we will start collecting an image. In this case, we can see that the image is fairly uniform. We see some vertical striation marks on the sample surface, but in general it looks to be a uniform area. So if we wanted to do a large area analysis, we could feel somewhat confident that this will give us a good representative analysis of the foil. Now what we want to do is, while using the SXI, investigate for different features on the sample surface. And so as shown here on the left, we can move the stage to an area that has a sort of a white blotch, and we will again use our SXI to see what shows up in the image. And very quickly we find that there are three areas of interest here. We have the somewhat uniform area across the middle. We have a dark area to the left that corresponds to the bright area we see on the photo. And we also have a sort of white blotch, small spot on the right side. So each of these looked like they could be interesting areas to analyze. And so by clicking directly on the SXI, we can recenter the image to our point of interest, and then we can choose to assign analysis points to those regions. So first we will click to save the image, and then we will just click directly on the SXI itself to set our analysis points. And we can label those as dark, bright, and average to correspond to the dark region to the left, the bright spot, and the kind of averaged gray area in the middle. Now, as you can see based on the scale bar to the bottom right of the SXI, for example, the bright spot to the right has a size of maybe 100 microns in diameter. And so we can choose our X-ray beam diameter related to the feature of interest. In this case, we'll use a 50 micron diameter beam. And we will do a survey across these 3 points simultaneously. So without moving the stage, we're going to collect survey spectra from these 3 points. And in doing so, the X-ray beam will simply be rastered to each of those points individually, and only that area will be illuminated, and the photoelectrons will be collected and added to our spectrum. And as we can see here in the blue spectrum, which is in the middle region of the SXI, we see a peak that represents most likely zinc of the metal foil. We also see some other peaks showing up. The blue spectrum is the one from the middle region. The red one is the black area. And then the sort of teal spectrum at the bottom is representative of the white spot. And so we can see that in the middle region we are detecting zinc. From the white spot we are detecting, the white spot is going to be your red spectrum. That has tin, which could be underneath the zinc. And in the dark spot on the SXI, again in that teal spectrum, we are seeing peaks for nitrogen, oxygen, and carbon, which could indicate that we just have an organic contaminant. And so in conclusion, what I hope that I've shown is that scanning X-ray imaging is a very powerful tool that we can use to quickly learn something about our sample and then use that information to help us determine how we want to perform our analysis, whether that is using a large area X-ray beam for a large averaged analysis across our sample, or we can choose to find small areas of interest and use a small diameter X-ray beam to find localized features to get a better understanding of some of the smaller features on our sample. And as we discussed earlier, because of the fact that the secondary electrons are collected through the same analyzer and detector as our photoelectron data will be collected, we have 100% confidence that the data that we choose, the analysis areas we choose on the SXI, will be exactly the areas that are analyzed for our XPS analysis. And again, for PHI XPS microprobe systems, SXI is a central part of the analysis workflow. So I want to say thank you for joining us for this webinar. And again, if you have any questions, please feel free to contact us. Thank you.